Okay, guys. So what I want to do now is to try to address some of your questions that you guys were still struggling with, what you guys were still confused about. And so the first thing that I have on my list here is isomers. So isomers share the same chemical structure. So hopefully you guys can see the writing on my little whiteboard here. So the easiest examples to use are glucose and fructose. So they both have the same chemical. Oh no, everything's backwards. Hold on a second. I think I can change that. Video settings. Mirror my video. Okay. Now I'm over here. Okay. So C6, right? So 6 carbon atoms, 12 hydrogens, and 6 oxygens. Now, the way that we put all those molecules together is going to determine the identity of it and what kind of isomer it is. So let me find my molecular little structure here. This is our glucose molecule. It has 6 carbons, which are those little black things, 6 oxygens, those are little red things, and 12 hydrogens, that's little white things. And I could take this apart and put it back together in a different way, using these same exact little ball thingies. So it would still have that same chemical structure, but it would be arranged differently. And so that's what an isomer is. And so the reason that we care about those is because they might respond and interact with our bodies differently. The easy example is fructose. So you guys have maybe heard about the argument, the disagreement, whether fructose is really bad for you or if it's just like because you drink too much sugar. So high fructose corn syrup, there's higher amounts of the sugar fructose that has the double bond in a different place than glucose. Our bodies have to expend energy to change that double bond in order to change the fructose into glucose so that we can use it for all ATP production and all that stuff. But there's an argument saying like, well, maybe you just shouldn't eat so much sugar. And then the other people are like, no, it's actually the fructose that's bad. So it's an area of active research, and there's not a consensus about it. So that's what isomers are. So the easy analogy would be, okay, if I gave, sorry, Cody and Nick, you guys are always my examples. I don't know why. If I gave Cody and Nick the same, like I bought 2 sets of Legos and I said, okay, here you go, put these together, you might come up with different structures. That's what an isomer is. And they matter because they're going to interact in our bodies in different ways. So somebody asked, what the heck are van der Waal interactions? Because I've put their name on the board and I didn't really say anything about them. Van der Waal interactions are very weak interactions. They're kind of like hydrogen bonding, but they don't involve hydrogen. They do it in different ways, but it's just a weak bonding. So I wouldn't worry too much about them. They're definitely not going to be on the test. Okay, cis and trans bonding. Why do we care? So here I'm going to pull out another one of my little molecular set thingies. So this is a fatty acid. Yay, autofocus. Okay, so we've got our double bonded oxygen up here. So this is our carboxyl group. This is what makes it an acid. And then we've got this big long fatty acid chain, all these carbon atoms. And so what you'll see here in the center is a double bonded carbon right here. And this particular one has that cis arrangement. Okay, these 2 hydrogen atoms are on the same side of the plane as this double bond right here. And if you guys will notice, when I let this relax, we've got a pretty significant little kink right there. And that's what makes it hard for these molecules to stack up nicely on top of each other. And this is what keeps it liquid at room temperature and as an oil. And so if I have to break these bonds. And let's see if I can actually do it. Let's see. So now they're in the trans arrangement, right? So here we're focusing on these atoms here between my fingers. And so now they're in the trans arrangement, where the hydrogen atoms are in different planes. Okay. And that actually gives us a more of a linear structure than the cis bonding did. So why do we care? Because kinkiness. So if it's kinky, it doesn't really stack up well to each other. Okay, this is a good one. Independent versus dependent variables, and where do we put them on our axes? So let me erase this. So the independent variable in an experiment is the thing that we change. And my NAU students today took their very first quiz. Oh, come on, autofocus. And we had a question on there saying, okay, the independent variable in our quiz example was the color of Skittles. And so we might have some blue Skittles. We might have some red Skittles. We might have some yellow Skittles. And we're trying to see which color the parrots prefer. What's the parrot's favorite color of Skittle? I don't know. It's a made-up example. It's not something that we would actually really do in science, but there you go. And so that was our independent variable. That's the thing that we were changing. We were giving the parrots different color Skittles. And then the thing that we were measuring, was how many of each kind of Skittle would those parrots eat. Now, that's the thing that we measured. So that's the dependent variable. The number of Skittles eaten depended on the color, right? So that's why we say it is dependent, because the number, so maybe they eat 5 or 10 or 15 or 20. And so the number of Skittles eaten depended on what color they were. Okay, so we're going to put our dependent variable on the y-axis. This is the crap we measure. And then our independent variable is the thing that we change. We put that on the x-axis. Okay. So somebody asked, nucleic acids. They're made of phosphate groups, some sugar, and some bases, and what else? Nothing else. It's just that repeated over and 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 over. Okay, it's just those 3 components that make up the nucleotide. The nucleotides are the monomers for nucleic acids. Phosphate, sugar, base. Over and 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 over until we want to die. Okay. Polar versus non-polar covalent bonding. What's that about? It's just how big of a pull do those electrons have toward one of the atoms or more. So let me pull out. Ooh, I don't know if I can. Okay. So using my little molecular model kit, we have our little water molecule. We have 2 hydrogens and an oxygen. And you can see, based on this little model, that the oxygen atom is the vast majority. There's more protons in the oxygen atom than there is in the hydrogen atoms. And so the electrons are going to, just by electrostaticness, just because positive and negative attract, the electrons are going to want to hang out around this molecule, the oxygen atom, a little bit more. And that means that the oxygen atom is going to have a little bit of a negative charge, and the hydrogen atoms are going to have a little bit of a positive charge. And so that's what polar and nonpolar is like, do you have a big fatty atom surrounded by little tiny hydrogens? And if you do, you are more likely to be a polar molecule. If we have nitrogen, so let's just kind of, so here's a nitrogen with two hydrogens. If we had it just like this, we would say, okay, yeah, the electrons from these two hydrogens are going to want to hang around this big, relatively big nitrogen atom. Now, if we had three hydrogens, oh, my Legos are not working. If we had three hydrogens on it, we would say maybe because they're kind of a little bit symmetrical, so maybe that wouldn't be a polar molecule. Okay, but for our class, I will tell you whether or not the molecules that we're talking about are polar or non-polar. So for us, that's not something that you need to be able to recognize, is this thing polar or non-polar, because I'll tell you if it is or not. Okay. Dehydration versus hydrolysis. This is a really important one for you guys to know. Because these are opposite interactions. So here is a molecular set of glycogen. So here is one glucose molecule. And so here's my glucose that I had before. If I wanted to add this glucose molecule to this glycogen molecule, I would have to find, I don't know if I can, I think these ones are glued together. So I would have to find, okay, here's where I stole that from, probably. So here's our oxygen and our hydrogen. And we would have our hydrogen, and we would need to pop off a hydrogen. I'll put that there. We would need to pop off a hydroxyl group, an OH group. Put that there. And then that means, oh, except for I need that bond. Now these guys can hook together. Okay, so now this, what I started out with a glucose molecule is now part of this polysaccharide. And that means that we have a little water molecule right there. Okay, it took energy for me to break those bonds and reform this other one. Now, so there you go. There's our dehydration, because we got kicked out this little water molecule. Now, if I wanted to break this apart, which I do because I don't want my boss to get mad at me for messing around with his molecular things, I need to break this. I need to water break this. So I'm going to add my water molecule back into here. Listen. Oh, that was lame. Sometimes you can hear them pop. Right? So that is the energy being released. And so now I can take this OH group from that little water molecule and stick it back on there. Right here, this is the one I messed with. And I can take this hydrogen and put it back on here. Okay, and so I'm using that water molecule to break apart a polymer into its smaller pieces. And so understand how those two reactions, dehydration and hydrolysis, are opposite of each other. When do you need to add energy? When do you get energy back out of it? Because those are going to be important things for you to understand. And if you do that biomolecule assignment, then you'll know what that means and how to do that. Okay. Okay, the function of cholesterol. So it's a precursor for a lot of things, including hormones, vitamins. It helps us digest our food and absorb the nutrients from it. So if you're low in cholesterol, you might not be able to get all of the nutrients from your food. With our Western diet, that's typically not a problem. Usually it's the other way around where we have too much cholesterol. So the other thing that it does that's really important is that it keeps our plasma membrane nice and fluid. If the plasma membrane is too rigid, then when the cells get bumped or whatever, it might actually break, and then all the cell juice gets released and leaks out. So keeping everything nice and fluid is one of cholesterol's job, because it kind of has this a little bit of a give to it because of its big, lots and lots of rings in it and stuff like that. Okay, cohesion and adhesion is a question that a few of you had. Cohesion, if you think of the words cooperate, corporation, coordination, the co part, right? That means working together. And so cohesion is water molecules attracted to other water molecules, right? So it's cohesion is samesies. Okay, water molecules hanging out with other water molecules, cooperating. Cohesion is water molecules cooperating. So hopefully that'll help you guys remember that. Adhesion is what makes water adhere to other stuff. So adhesion is water molecules forming hydrogen bonding with other substances. And so remember, we talked about water movement through trees, and we did the little capillary tubes where the water molecule was actually being attracted up the side of the glass. So add, others, co, self. Okay, cohesion is water molecules cooperating. Adhesion is water molecules adhering to other things. Sterols versus steroids. I just realized why everybody's confused about this, because I made an error in the PowerPoint. Steroids are the larger group of these lipids that have these carbon ring structures. So there's 4 rings, made of carbon, and sterols have an oxygen group on them. And so maybe just try to do some Googling of some images of those for you. Phospholipids. Phospholipids, I think, are the coolest kinds of lipids. It might even be my favorite kind of biomolecule because they're so cool. Because they're one end, they've got a hydrophilic end. I don't know if you can see that. Come here, camera. Okay, there's a hydrophilic phosphate group. Ooh, that's terrible. And then, so here's a carbon, here's a carbon, here's a carbon. We'll draw them like this because that's how the fatty acids usually are. Maybe we've got a double bonding here, and so this one goes out that way. Okay, oops. You can't have 2 things attached to you. What are you talking about? So we've got a hydrophilic phosphate group, and then we have hydrophobic fatty acid tails. And so this is going to want to, this part of the molecule is going to want to face the watery environment, and this hydrophobic fatty acid tails are going to want to get away from that. And so what we see is when we have a whole bunch of these things together, they kind of start to form. You guys have seen the droplets of oil in the water. And that's what it is it's the fatty acids trying to get not to interact with the water. Get away, you're really scary. Whereas the hydrophilic's like, hey baby, how you doing? I want to hang out with you, water, because you're super cool. All right. So that's why I like those. Omega-3 fatty acids have multiple double bonds. And they're important because we lack the cellular machinery to produce those ourselves, and so we have to eat other things. I think I answered a question on Canvas about this, kind of talking a little bit about how aquatic insects actually have a higher omega-3 fatty acid content than terrestrial insects because they're eating algae and stuff that are better at making omega-3 fatty acids. And that's why we often see, say, oh, you got to eat fish, you got to eat tuna and salmon to get your omega-3s. It's because they're there eating those aquatic insects that are eating the algae that make omega-3s and omega-6s. Okay, so hopefully that answers and clears some stuff up for you guys. I'm gonna try to make myself as available for you as I can tomorrow. I do have a lot going on, so I'll keep watching the Canvas email. So let me know what questions you're coming up with and where you're getting stuck. Okay, good luck on the exam, and I'll see you guys on Thursday.